Sir, please begin.
Okay, sir. Good morning, everyone. Respected Dean, sir, HOD, sir, my senior faculty members, my colleagues, and my dear friends. So today we are here to present a topic on engineering tissues for transplantation. And we have with us our speaker and guest, Dr. Naresh Kasoju. So let me first introduce about Naresh Kasoju, sir. Naresh Kasoju is presently working as a scientist C in the Department of Applied Biology, Biomedical Technology Wing at Sri Sitra Tirunal Institute for Medical Sciences and Technology, Kerala. He has obtained his PhD from IIT Guwahati in biotechnology. He has also postdoctoral experiences from the Institute of Macromolecular Chemistry, that's Prague, Czech Republic, and also from the University of Oxford, UK. His research interests include mesenchymal stem cells and exosomal-based therapeutics for regenerative medicines application and 3D bioprinting, silk fibrion, and other natural polymers-based biomaterials for biomedical and other areas. He has published more than 50 articles with around 3,000 plus citations and I-10 index of 29. He is the editor of 3 books, guest editor of 2 journals, special issues, and editorial board member of 2 journals, early career editorial board member for 1 journal, and reviewer of around 50 plus journals. He and his team has presented work at several national and international conferences and received quite a few awards. He filed 6 Indian patents and 3 Indian designs registrations so far. He has received a total of 9 research grants from internal and external agencies, including DST and SERV. He is the member of the prestigious National Academy of Science, India, and Royal Society of Biology, UK. So please join with me in welcoming our guest and speaker, Dr. Naresh Kasoju, sir. Sir.
Yeah, good morning to respected faculty members, dean and head of the departments, and dear friends. Thanks for the wonderful introduction, Hemanga. Hope I'm audible and the slides are visible.
Yes, sir.
Yeah. So, at the outset, I would like to thank Hemanga and all the department colleagues for having me here today. And so today I'll be mostly talking some fundamentals and basics of tissue engineering field for the benefit of general audience. And I would also touch upon a little bit of what work we do here. So I will start briefly about my institute where I'm working. And then I will give you about aspects of tissue engineering, especially what is the need for tissue engineering and what are the concepts available in tissue engineering field, including the cutting edge or the latest trends in 3D printing and 3D bioprinting. I would also give you some details on cell material interactions and how these interactions influence the fate of tissue engineering applications. And as I said, like I'll give you some idea about what sort of research that we do here in our institute, and with a little bit of future prospects for all of you people, the young people. So the Institute, Sri Chitra Institute for Medical Sciences and Technology, it is an institution of national importance and an autonomous institute under Department of Science and Technology, Government of India. So I can say that it is very much like a AIMS level institution. Of course, we do have a hospital which is specialized in cardiology and neurology. And then we have a biomedical technology wing, which is an R&D department, which I'm working in. And then we also have a Achyuta Menon Center for Health Science Studies, so which is looking into public health aspects. So this is a very unique combination of R&D, a hospital, and the public health department under one umbrella. And this is a 40-year-old institution having this setup of material synthesis to almost to the level of clinical trials under one platform. And that is why it has achieved a great success in bringing up a variety of medical devices. So previously, the cardiac surgeries or any other heart valve related operations used to cost a lot of money, because it involves imported heart valves and other associated devices. So this particular institute was on a Make in India mission for the last 4 decades, and this institute has been instrumental in developing indigenous medical devices for the last 4 decades. And as you can see, the first and foremost product which has come up from this institute was artificial heart valve, which is also known as the Chitra heart valve. So from 2 to 3 lakh rupees, the cost is now cut down to 20,000 rupees because of indigenization. And also it is implanted in millions of patients, and all of them are happy. And even the first patient who received this heart valve 25 years back is still healthy, and the valve is still functioning in that. So we do and then you have all other associated instruments which are required in the cardiac surgery. And then there are also other materials based on neurological conditions like hydrocephalus, shunt, bioceramic composites, and dental products, and other vascular related grafts. So as I said, this has a all-in-one sort of a infrastructure facility, and getting a product of this high-risk products into the clinical setup requires a series of laboratory setups which are highly accredited or highly what do you say comply with international standards. So our labs, starting from material synthesis, including physicochemical characterization, and then design analysis, including the in silico analysis, and then you have animal related studies, small animals and large animal, and then the product release study. So we have a setup of laboratories for all the aspects of product development, and these laboratories are accredited by agencies like NABL, which is based in India, and agencies like COFRAC, which is based in France. So we have this setup, and eventually over the time we realized that these the unique facilities that we have also can be shared with others. So the institute has also started sharing the facilities in the form of consultancy programs. So any other companies or universities who are interested in biomaterial development can come to us, and we can perform the work on consultancy basis as a service to that particular commercial or the customers. So that's briefly about the institute. And coming to the topic of organ failure and transplantation, or the tissue engineering, why exactly we need to look into the aspect of tissue engineering is because we see on day-to-day basis in the newspapers and TVs that a lot of cases of organ failure, including a very serious death cases because of various reasons. So be it accidental injuries leading to tissue or organ failure, or disease based organ failure, for instance COVID. So you have a lung defect or lung failure, and subsequently multi organ failures. And then you also have age related failures in the form of tissues and organs. Okay, now the current gold standard approach is obviously the tissue and organ transplantation. So where we depend on a donor, and if the donor can actually donate all the organs and tissues, so he can save, or he or she can save up to 8 lives by donating various organs. And he can also, or she can also save up to 50 patients through different tissue donations, like corneal tissues or skin tissues or any other not organs but the tissues. Okay, but unfortunately, like we can also question our own self, like how many of us have really signed for organ transplantation. So it's very poor. So if you look at the numbers of organ donation rate persons per million, so the European countries like Spain have a very decent number, and followed by US, and then many other Western countries. So they have a very good rate of organ and tissue donation rates. But unfortunately, when it comes to Indian situation, so the awareness is very poor, and we do not have that much organ donation programs. So the bottom line is that we do have a very serious gap between the demand and the supply of these tissues. So unfortunately, like if you search, there are no other products or other alternatives for this tissue level transplantations. So realizing this potential, so it was a development, interdisciplinary fields. So the developments in the field of cell biology as well as material science have converged 2 decades or 3 decades back, and led to the formation of a new branch of science or engineering called tissue engineering, wherein this picture here has actually revolutionized the field, wherein you can see that there is a rat and with a human ear on its top. So this is nothing but a scaffold, a material which is seeded with cells on it, and then which was transplanted on the back of this rat, and it has anastomized well that it become part of that particular animal body. So I'll tell you exactly what that particular process looks like. But then there are three main components in this tissue engineering process. It's called tissue engineering triad. So you have a scaffold, or which is a template basically. So it's a material which is made into a particular shape or size depending on the tissue of interest. And then there are the second element is the cells. So depending on the tissue of interest, you take cells of that particular tissue, and then you also have biological factors which are required for an optimal cell response in that particular scaffold. Then you have no you have three factors: cells, biochemical factors, and then you have scaffold of interest. So how do we use it? So these three elements are actually used in different combinations and permutations. So there is no one protocol that you take cells and then this and then that. So people have tried multiple ways of using just the scaffold alone, or just the biochemical cues alone with the scaffold, or cells with scaffold, or cells with biochemical cues. So you can try different combinations and permutations, and they can be first developed in vitro to develop a mature tissue and then go for transplantation in vivo, or they can also be used in combination directly in the in vivo situation using the body as a bioreactor. So there are again multiple possibilities in it. But having said that, a typical conventional tissue engineering process is a kind of a cycle. So you start with the patient, and then let's say you want to treat the cartilage defects in this particular patient. So what you take a biopsy of a healthy tissue in that particular region, and then you try to isolate the cells of your interest, and then you expand it to the required number. So I require, let's say, some 10 million cells. So you isolate few and then try to expand it to 10 million scale, and then you develop a matrix or a scaffold, and then you seed your cells of interest on top of that particular scaffold, and then you incubate in vitro in order to mature that particular tissue to a desired function or a desired structure. And once that is matured enough, you go back to the patient and then you implant in the clinical setup. So that's a typical flow of work in tissue engineering field. So I'll briefly touch upon various these three components, that is cells, materials, and biochemical factors. So when it comes to cells, so we do have multiple options. So we can directly go for a tissue derived somatic cells. So if you are interested in liver, so you go for directly take cells from the liver, and see if you can use it directly. So the best possible source is obviously the autograft. So because you don't have any immunological problems, but then it's not always that you get a good number of healthy cells from that autograft. So for instance, you have a patient who has a burn injuries, but none of the skin is healthy enough to isolate cells. So in that case, you have to depend on somebody else. So if you have an identical twin, so that is again another ideal source, because you will have relatively less immunological complications, because the genetic genotype is more or like comparable. But again, we don't have twins all the time. So then you come to an allograft situation, so where you take up somebody else who are not directly related, uh, given that certain immunological parameters match with each other. And there are also situations, and in fact recently you may have seen, like animals are being used, or the tissues from animal sources are also being used for transplantation application. So but that's a rare thing, so because you have a lot of immunological reactions coming from these xenografts. But allograft is a kind of gold standard approach in the field of tissue engineering. But then you may not really have the somatic cells enough, number one. And number two, even if they may not be having a ability to proliferate. So if you take, for instance, hepatocytes or neural cells, so you can isolate cells, but then they do not have that proliferation ability. So if you want to develop a scaffold or a tissue of liver, and then you want to actually have some 10 millions or 100 millions in the lab, so they may not able to proliferate. So people have started looking for alternative cell sources, and the most obvious choice other than somatic cells would be the stem cells. So, and as you may have already learned in your basic courses, so the stem cells are those magical cells which have some self-renewing ability, as well as ability to differentiate into multiple lineages. And they come from varieties of sources. So they can be embryonic stem cells, which are derived from embryonic developmental stage. So they have a totipotent and pluripotent capacity. And then you also have some fetal derived adult stem cells, so for instance amniotic fluid derived, and then you have umbilical cord derived and other tissues. And then you have adult stem cells, which are tissue derived. So you have bone marrow, blood, adipose tissue, dental pulp, and etc. So you can isolate cells, stem cells from those tissues as well. And the new concept of pluripotent stem cells or induced pluripotent stem cells is also on the rise, because of the fact that you can take any part of human body, cells from any part of human body, and you can convert it into a stem cell, and then de-differentiate or differentiate back into some other desired cell type. So various sources or various stem cells are being explored widely in this field. And of course, every source has some advantages and some disadvantages. For instance, the embryonic level, we have very strict ethical regulations and standards in that case. So cells from umbilical cord are easy to isolate, and they are having less ethical concerns. And somatic cells, you do not have any ethical concerns, but then there is a potential of tumorogenic tissue formation in some situations. So everything has its own plus and minus. So we have to assess the advantages, risk benefit, according to that. Accordingly, we have to take a particular source. The second element of tissue engineering is the biochemical factors. So of course, as you know, the cell is a fundamental structural and functional unit of life. So it can actually survive on its own. But then we are multicellular organisms, and we have different levels of organizations in our body, starting from cells, and then you have tissues, then you have organ systems, and then complete body. So at each level, because of the interactions of cells with other cells and the extracellular matrix, it has evolved different systemic functions, okay, which are not at the cellular level. So that is possible because of various communications, and those communications are basically 6 types, and some are based on soluble factors. So for instance, there are mechanisms wherein the cell has intracrine signaling mechanism. Basically, you have a growth factor or a chemical factor which is not really released outside. It is produced in the cell and it is used in the cell because of in various pathways. So those are intracrine signaling molecules. And then you have some autocrine signaling systems where the cell is releasing a particular molecule outside, and it reacts or senses the outside environment, and then after modification it is being taken up by the cell in order to do something. And then you also have very famous paracrine signaling and then endocrine signaling. So these are based on the soluble molecules. And then you have two other interactions which are non-soluble in nature, and they are direct interactions. So a cell which is next to another cell, they would communicate through these cell-cell interactions or cellular junctions, let's say. And then you also have an extracellular matrix, and the cell will interact with that extracellular matrix in order to sense what is going on outside. Okay, and if you see, look at the typical transduction or a signaling pathway. So you have a cell which produces a signaling molecule, and it is being received through a receptor of a target cell, and then the signal is transduced through primary and secondary signaling messengers to the target gene. Either it gets activated or it gets suppressed, and then you have a biological response accordingly. Okay, and these signaling mechanisms and these biochemical factors does a lot of things, both positive and negative reactive reactions. So it can aid in cell proliferation depending on the requirement, and it can also tell the cell to die. Okay, so you have this apoptotic self mechanism where there is a suicidal mechanism. So the cell can differentiate into a particular tissue of interest, or it can also de-differentiate. So you have something called epithelial to mesenchymal transition. So where a somatic cell or a differentiated cell is being de-differentiated back into a stem cell related state. So technically, it can actually heal a wound, or it can also lead to a wound. So it has both positive and negative. So it is a very these are very critical in the in maintaining the homeostasis, and we need to really understand all these mechanisms in order to maintain the homeostasis. So some of them can be encapsulation, some of them can be non-covalent binding, and some of them can be covalent bonding. So the first one, if you look at, is physical absorption. So you simply coat your material or a scaffold with a solution containing a growth factor of interest. So just because of the surface level charges or interactions, it might just simply absorb on the surface. But then these interactions are very weak. So again, if you simply wash it, they might get released. So this will lead to a very poor binding and very quick release. You can also have ionic bonding based on the ionic charges. So if your surface is positive charge and you use a growth factor which is negatively charged, so you have a ionic interaction there. You can also have a affinity binding method. So where you have some functional groups on the surface of your material, and these can be have some special affinity towards the growth factors of interest. You can also have a matrix entrapment where you can entrap the growth factors in a network of hydrogel. You can also have covalent bonding directly through some chemical cross-linking methods. And then you can also have interpenetrating networks of cross-linking where you have a high load or a high reservoir of growth factors. And there can be a simple encapsulation systems where you just load a solution in a membrane-like sac, in that particular thing. So different things have different mechanisms of attachment, and each thing has a effect on the release kinetics. So for instance, physical adsorption is quick release, whereas the covalent bond interactions require some special agent to cleave that covalent bond, then only it will be released. Okay, so depending on the chemistry of your material as well as the growth factor, plus depending on the kinetics of release, you may have to choose a appropriate method. Coming to the third aspect of tissue engineering, that is the material part. So biomaterials are, it can be the natural or synthetic systems or materials. These are introduced into the body as part of a medical device, or which is used for tissue replacement. You can also have a varieties of materials here. Basically, you have metallic materials, then you also have polymers, which are derived either from synthetically or it can be isolated from natural sources. Then you can have ceramic materials, which are bone like substitutes, and then you can have a composites, which are a combination of metals and ceramics. So again, these biomaterials vary, and they will have different merits and demerits, because a metallic implant versus a ceramic implant versus a polymer, so they will have different merits and demerits, and not one single material cannot be used for all applications. So depending on the tissue of interest, you may have to choose a material of appropriately. So for instance, if you go for a contact lens related material, so contact lens have to be transparent enough, it has to be tough enough, but it has to be soft also. So you have to choose a material according to that requirement. You cannot make a film which is translucent, so the whole purpose is not going to be accomplished. Then if you are going for some bone related application, so and the bone related is like it is a basic function is to load bearing function. So there is no other metabolic function. So you can take metallic implants in that case. Okay, and if you're talking of wound healing, so again the requirements of that particular tissue will be different. So depending on the tissue of interest, you may have to choose a material accordingly. But then there are certain basic things which have to be met. So first of all, it has to be non-cytotoxic, because cell is the fundamental unit, as I said, and any material which you use, it should be non-cytotoxic. It should be non-thrombogenic as well, because when you cut, the first thing that comes out is the blood. So your material should not induce any thrombogenic response. It should also not elicit any immunological response. Okay, plus if it is bioresorbable, it will be desirable, because you want that material to degrade in the body over time, and it will be taken up by the native tissue. And it should also be bioactive. So instead of just the material alone, which takes a lot of time to induce a particular response, you functionalize by including certain growth factors of interest. So it becomes a bioactive. So the wound healing or any other engineering tissue will become more fast. And then there is also bioresponsive phenomena, which is recently introduced, because you load a growth factor, but you don't want to waste that growth factor when there is no need. So you want that growth factor to be released only when there is a need. So the people are trying to develop smart materials or responsive materials, where you can, so depending on the pH, let's say, you can release that growth factor, or depending on the stress or mechanical factors or any magnetic field. So depending on various physicochemical responses, the growth factor may be released. And as for so those are the three elements: so cells, materials, and growth factors. And when it comes to the process, the actual practical approach of doing a material, I think, platform. Okay, so when you actually look at the process of tissue engineering, there are two approaches. So the first one is the top-down approach, and this is the conventional way of doing things. So where you have a scaffold which is typically made of polymeric materials, and this is being used as a structural support for cell attachment. So you have a spongy like a porous scaffold here, and you take cells of interest and you directly seed on top of that particular porous scaffold. And when you culture in vitro in an incubator, so these cells will get seeded on top of that pores, and they will gently go deeper into the pores of that particular scaffold, and they would proliferate, and they would try to form a three-dimensional tissue according to the shape of that particular scaffold of interest. Okay, so here the pore properties are very critical. The pore diameter, the pore volume, and the pore pattern, and the interconnectivity. So whether the pores are interconnected or not. So this will influence the overall fate of tissue engineering there. And also some surface features like roughness, surface chemistry, wettability, and surface energy, because the cells, when they seed on top of it, so depending on the surface features also the response will differ. And certain other features like mechanical properties, like if you're talking about skin, so it has to be like little elastic or little soft in nature. If you talk of bone, it has to be very tough and rigid. So accordingly, the material properties have to be optimized. And what about the form or the shape? So again, depending on the tissue of interest, you have to convert your material into various forms. Okay, so for a soft tissue material, so you may need a jelly like material. And for any other patch, if you want to make a skin dressing, or if you want to make a cardiac patch, so you can develop a material which is having a nanoscale fibrous architecture. So for corneal related applications, you can have films which are transparent in nature. So for other, maybe if you want to fill up space, you can have a micro scale particular scaffolds. For tissues like blood vessels or neural grafts, so you need tubular structures. And for any other conventional tissue, so you may want to use some porous spongy like material. So again, you can process into various forms, but then there is nothing universal here. So people will explore different scaffold fabrication approach depending on the requirement and depending on the the material properties. So not all materials can be processed into all the various forms. So you, depending on the material and depending on the tissue of interest, you take a scaffold fabrication of choice to develop a scaffold of interest. The second approach is the bottom-up approach, which is the reverse case. So you start building the tissue, one cell at a time, or it's like constructing a home by brick. So in the conventional way, as I said, you have a scaffold and then seed the cells on it. But in this case, you start with the cellular aggregates or cellular sheets or some small cell-laden gel-like materials or beads. You assemble these aggregates in a particular shape or size, and then they fuse together or self-assemble together to form a tissue of interest. Okay, so the scaffold based technologies or the top down approach, so you seed 100 cells on top of a scaffold, but not necessary that out of 100 will seed 100 out of 100 will seed there. So that was the problem with the conventional top down approach. Whereas in the bottom up approach, because you are encapsulating in a gel, so 100 out of 100 will remain in that particular tissue of interest. You can fine tune the mechanical properties, and you can also have a self assembled tissue architecture. You combine different cell types, and you can actually develop a cell tissue or a tissue architecture according to the native tissue, which is otherwise not possible in a conventional top down approach. And you can also incorporate a variety of growth factors in it. Okay, so the bottom up approach has thumbs up for all the various features that we require. Okay, So one of that bottom-up approach is 3D printing, which is also called additive manufacturing. So in the conventional manufacturing, which is a subtractive manufacturing, you start up with a material and then you start drilling out the unwanted material in order to get an object of interest, but you end up with a lot of waste in it. So whereas in the additive manufacturing, so you have start with a raw material and you do a design and start printing layer by layer in order to get an object of your interest. So you have a very less waste here, but the most important thing is that you can develop materials with a lot of intricate designs. Okay, so that is the advantage of additive manufacturing. So this is a conventional 3D printing. So when you say 3D bioprinting, the difference is the bio aspect of it. So in a conventional setup, you have a print head, which is simply heating a polymer filament at about 100 to 200 degrees, depending on the polymer. And once it is molten, so it just comes out through a nozzle and you collect it on a stage, which is controlled in XYZ directions. But in terms of the bioprinting, you first of all have a sterile environment because you are dealing with cells, and then you don't heat at 200, rather you heat at 37 degree, which is a physiological temperature. And then in the syringe, you don't have a polymeric plastic filament, rather you have something called a hydrogel system, which is called a bio ink. So where you keep your cells of interest in the gel format, and the piston is pushed pneumatically by using air pressure, and the gel comes out through that particular nozzle and gets collected on the stage, which is again controlled in XYZ directions. So it's a simple setup. It's just that when it comes to bio, things get a little complicated but also interesting. Okay, so in terms of printing modalities, so you have three bioprinting modalities. So the first one is an extrusion base. So you simply have a syringe loaded with a bio ink, and then you start pushing the material either by pneumatic or by piston or a screw based, and you end up having a filament coming out of that particular nozzle. The second one is a inkjet printing, where you have a piezoelectric or a heater based transducer systems, where you have a droplet sort of a deposition coming out of. And then you also have a little sophisticated one, which is called a laser assisted printing, which gives a better resolution compared to the extrusion based printing. So this is a typical setup of a 3D bioprinting, as I just mentioned. So you have an aseptic environment, and then you have a print head, which is holding a syringe containing a bio ink, and then it is pushed either pneumatically or manually or mechanically, and it is through a nozzle, it comes out and gets collected, and the whole machine is controlled by a computer. So the computer and the stage, everything is mechanical here, except and it's all automated, and you have very good control over all other parameters except the bio ink. So the most critical component of the entire 3D bioprinting is the bio ink, which is nothing but a hydrogel. So it should be optimized for printability aspects. So we should be able to extrude a very nice filament out of it without any beads or anything. And when you print it, it should have a shape fidelity. So you should have a very nice rigid three-dimensional structure out of it. It should be having some growth factors to promote the cell differentiation, adhesion, and proliferation, etc. And as I said, it should also be mechanically strong. So when you say gel, it should not be just like not so weak. So you should still be able to handle it properly. So yeah, so this is what I mean by printability. So when you drop any liquid, so usually you see a droplet kind of a feature there, but in bio ink we should have a very nice free flowing sort of a filament coming out of that nozzle. Okay, so but when you free flow, again the chemistry should be optimized in such a way that once it comes out of that nozzle, it should immediately gel, so that when you print one layer and then second layer on top of it, they should not fuse together. It should be self-standing. So that should not so the filament should be, as soon as it gets out of the nozzle, should be cross-linked by some means, either photo-based or chemical-based, so that it becomes gel, and the next filament which you print on top of it will be self-standing without fusing. So the biocompatibility aspect also, as I mentioned, so it should be compatible with the cells, and you can include growth factors as required, and it should also be able to mechanically strong so that we can handle very easily. And another aspect is that it should be able to biodegrade over time. So that's about the both bottom-up approach and top-down approach. And when it comes to what determines the entire fate is determined by how the cell is interacting with that particular material in that three-dimensional context. And there are various factors which influence that relationship. So whenever you have a foreign object and you don't accept it immediately in your home. So similarly, whenever you have a foreign object which is a medical device or any other object, whenever you try to implant in your body, that you have to go through a lot of tests. Okay. And varieties of features. So for instance, the morphological features, the nano scale topography, the surface chemistry of that particular material, the mechanical properties, and other biological features, they all will influence the cellular response. Just for an example, so there's an interesting paper published in 17. So you have what is known as endothelial cells, the cells which make blood vessels. So when you culture in a 2D plate versus a 3D hydrogel system, you can clearly see the difference. Okay, so in the conventional 2D culture, the cells are just attached and they are not forming any tissue level organization. They are intact on their own. But whenever they are cultured in a three-dimensional gel-like structure, gel-like scaffold, you can see that they start to self-assemble and start to form the blood vessels of interest. Okay, so the conventional versus the 3D culture, so it makes a big difference in the tissue function or the cellular response. Similarly, whenever you culture cells in a defined chemical organization, so this is a material which is functionalized with RGD peptides. These are recognized by cells for anchorage. And whenever you define or when you pattern that in a different shapes, you can see that one same cell, a stem cell, which is seeded here. In this case, so a shape of this star is inducing an osteogenic differentiation, whereas a spherical shape is inducing a adipogenic differentiation. So depending on the pattern also, you can induce a specific cell response. Okay, because the stress fibers which are acting in this star shape versus the spherical shape will be different, and those differences will have effect on the molecular network or the molecular pathways, and then accordingly it can either induce an osteogenic response or adipogenic response. Okay, so and also the substrate stiffness. So we are talking about a hydrogel, but what stiffness it should be, whether it is a soft gel or whether it is a stiff gel, depending on the rigidity of that particular hydrogel, the cell can be differentiated. So if you are talking from 1 kilopascal to 100 kilopascal range, so one cell which is seeded on that soft gel can differentiate into a neuronal type, whereas if the gel strength is more, about 100 kilopascals, so the same cell will be able to or promoted towards a bone related regeneration. So we have to pay attention to these sort of chemical and mechanical or other properties of the material. So I won't go much deep into that, but that's just to give you an idea that the material properties would influence the cellular response. So coming to our work here in our campus, so we have a 3D bioprinting program, and we are working on skin and liver related applications. So this is just to show that the skin has a dermal and epidermal layer, and dermal layer is mostly with matrix with a few fibroblast cells here and there, and then you have a epithelial layer which is composed of keratinocytes. So this is a example of 3D bioprinted gel. This is a single stack and this is a multi-stack, as you can see. So we can print multiple layers on top of each other, and you can also print any shape of your interest. There are multiple aspects of characterization. So this is what I was telling you, that we should have a smooth filament coming out of that particular nozzle. And we there are things like velocity that we should control. So when you print it, it should exactly match with the design that you give. It should not collapse. So there's also a filament collapse test which is performed. We also print it in different shapes so that we know what is the kind of angle that we can get. You can also assess the line space. So what is the space that I should give between one line and the other line? And we also do some other calculations to see what is the pore size which you get out of the printing process, what is the smoothness of the filament, and what is the crossover fusion at that particular structure. And we also do a swelling study. So once you incubate in a medium over a time period, so what is the level of decrease in the pore size, and a lot of other calculations. And some in vitro studies. So this is a micro CT, and you can see some live dead staining, and you can also see some dual staining and HE staining here. So it is looking like a native tissue, like skin. So you have a dermal layer with the fibroblasts, and then you have a very thick, a densely populated epithelial layer. And we did in vivo studies as well. And you create a wound, and then you implant or transplant your 3D bioprinted skin graft, and you close the wound with the surgical sutures, etc. And over a couple of weeks, we could see that the wound is totally healed. We also do some other applications on corneal related, and this is a bottom up approach again, and it is called a cell sheet engineering. So cornea, as you know, it is a transparent outermost tissue, and then epithelium is the outermost layer of it. So what we do, we develop a tissue culture dish, and we coat it with a special polymer which is thermoresponsive. So we culture cells on it, and once the cells are grown, we decrease the temperature of that particular dish. And by doing so, in the process, the polymer changes its phase, and the cellular monolayer loses its contact with the substrate, and it comes out as a sheet. So this is how you see, like in a 37-degree, you see a very good monolayer of cells. And as soon as you decrease the temperature, the cell monolayer comes out as a sheet of it. And some other examples of silk based materials. So silk, as you know, they are used previously as a suture materials long back, and they are biocompatible in nature, and you can process it in the form of a liquid, and you can see a nice film which is optically transparent, and it's also flexible, so you can twist and do any sort of thing, but it still retains its shape. We also develop tissues based on scaffolds which are made from electrospinning, and we do co-culture systems. This is an example of a skin tissue where you can see there is a dermal layer, and then you have an epidermal layer, and this can be packed and shipped also. We also work with bioreactor systems where we have a bone tissue engineering project where we seed cells on a ceramic graft, and they are kept inside a microwell plate or a multi-well plate, and they are cultured in a dynamic condition in order to get a three-dimensional tissue. So that's about our just a glimpse of our work. And but when you talk about translation from bench to bedside, of course we have the challenges in the R&D level, but also there are certain agencies called a CDSCO, and these are the agencies designated by Government of India to check whether the product development is as per the standards and as per the laws of the country. So there are different aspects to it, and these also have to be well thought in the initial stage of R&D itself. Okay, because initially we do research just based on literature search and other things, but then we, for the sake of getting a good publication, we try to add a lot of things to it. But then when you look at the commercial aspect of it, so the more you add in your process, the more costly it becomes and the more complicated it becomes for the approvals. So you make your research simple so that you get your necessary approvals in a much easier fashion. So it's not a theory, it is actually practical, and there are commercial products available on the market. So there is a Appligraph company which is doing artificial skin in States, and in Europe they have recently got a approval for a corneal epithelial derived tissue fabrication, and you may have seen recently that a 3D bioprinted ear was transplanted onto a patient in Western world. So these are live examples of commercialization. So it's not just a fancy science or engineering. It is a very useful technology for healthcare, and it would also become very personalized and precision, because the more you introduce machines to it, and the more introduce you technology to it, it becomes more precise, it becomes more personalized. So the roadmap is again, like, you have to have a lot of collaborations for industry level and consortium level collaborations, and you have to follow standards and know what is required by law, what is required by international guidelines, and then you have to promote that space for innovation. And in terms of educational systems, so we have to inculcate or include programs both theoretical as well as practical level in this particular aspect, so that you get that necessary training from the basic education courses itself, because these are things which are going to really take much of the market, as you can see. So from 2026, so the increase in the overall revenue, or the increase in the overall budget, is almost like 15%. So there is immense scope. So it is currently at 9.8 billions, and the market is expected to increase by 26 billions by 2026. So we are very in order to get good opportunities, so the field requires a lot of people. So irrespective of your background, whether you are a biologist or a chemist or engineer or mathematician or business, or you are interested in IP or quality and regulatory affairs, there are immense opportunities in this field. So with this, I think I'll just conclude here, thanking my institute and the funding agencies, and also to the the organizers. Thank you. And if there are any questions, we'll be happy to interact.
Thank you, Naresh sir. Thank you for such a lovely session. Yeah, here are sir for 5 queries from our students, which are written in the questions and answers column.
Yeah, I can see. Yeah.
Yes, sir.
So is there any material available used in transplantation of stem cells? If someone has positive, doctors generally suggest bone marrow transplantation. So, well, bone marrow transplantation is a different thing. So they don't use any material there. They would take the bone marrow and directly infuse at a site of injury. But there is no as such a universally accepted material per se for such applications. So it all depends on what type of tissue we are targeting, and depending on the tissue of interest, we may have to take a material with the necessary physicochemical properties. Do prosthetics also come under tissue engineering? If it is without cells, so technically it is out of tissue engineering, because we are working with cells. So then only it becomes, so anything without life is a prosthetic, to my understanding. So if you have cells in it, and if you want to make it biologically responsive, that is where tissue engineering comes up. Are there any biological cross-linkers instead of cross-linking polymers like acrylamide? Yeah, I mean, there are, when you say cross-linking, even an enzyme, which is a biological origin, can also be used as a cross-linker. So there are multiple options for cross-linking. Can hydrogels work for in vivo tissues? Well, it's a generalized question, and the answer can be also generalized, yes. It can work, but again, depends on which tissue we are targeting. Okay. So hydrogel may not work for load-bearing applications like bone tissues, right? So it cannot withstand. So depends on the tissue, basically. What are some aspects that can go wrong during tissue engineering process? In fact, In terms of material, so as I mentioned, like the pore properties of the scaffold are very important, and the mechanical properties are also important. And in terms of the way the cells respond to the material, so these things have to be studied well in order to understand and in order to get the desired outcome. And in terms of process as such, I don't think there are much to worry about. Yeah, I think that's all.
Yes, sir. Thank you, sir.
Yeah, any other questions for anybody?
Anyone?
Yeah.
From the student? Is there anyone? Any query? So your speech was very much clear. I think it is very clear to the student. The simple and so nicely you have understand taught us the things, materials. Thank you, sir. Thank you very much, and hope we will see you in future. So in Parul University, on the behalf of Parul University, I like to thank you from the bottom of my heart for giving us the time and enlightening the students with such a lovely and important topic. So first of all, I like to tell about the Parul University. That Parul University is West India's best private university, and it is also ranked among the top 50 private universities in India for leading innovations. It is awarded as the best private university in placement with around 35,000 students and 2,000 faculties. We have around 2,000 plus international students, sir. So hope we will see you physically here in such event in your about future. Thank you, sir. Thank you very much.
Yeah, sure. Thank you. And best wishes. Yeah.